I think I have explained it. If I'm not wrong. Alia, so did I explain these all things in last class? That I don't know. We did till this point. That I am confident. So have you noted down all this part, all these factual things, or rather factual things, all these things? I'm not audible. You're not audible to me. Ali, I didn't got it. Your voice is not uh, audible to me. Can you text me?
Yes, sir.
So can you text me? Had I explained this point or not?
Yes, sir. You already did this.
Okay. I already discussed. Fine. So this next thing, which is as per your NCRT, that is that I need to discuss with you. What is the gram positive and gram negative bacteria? This is important for your school examination. Right. That is very important. What is the gram positive, gram negative? You must have listened this term gram positive and gram negative bacteria. Look, the gram staining. This is a practice. This gram staining discussed and it is discovered by a scientist named Christian Gram. Since Christian Gram in 1884 discovered the practice of staining bacteria. And that's how he demarcated that these are the gram positive bacteria, these are the gram negative bacteria. So what he did, he taken bacteria, and after taking bacteria, he stained these bacteria with the help of a dye called crystal violet or gentin violet. This is the name of that dye. So he stained these bacteria with the help of crystal violet or gentin violet. Uh, this is weakly alkaline dye, right? Bacteria get blue color. So bacterial outer cover taken the blue color. Now after this step, the next step is they treated the bacteria with 0.5% iodine solution. After treating these bacteria with the 0.5% iodine solution, they wash these bacteria with alcohol or acetone. After washing those bacteria which retain blue color, they are called gram positive bacteria, and those bacteria which which become colorless or they lost the color, they are called gram negative bacteria. Is it clear? Clear to both of you? Keep on telling me. Please draw this flow chart, then I'll explain. Great. Please note down this chart quickly, as it is. And this is 3-marker question. Very, very important question for your school examination. And when you get this question, first you have to draw this flow chart. Then you have to write it down. Let me know when you note it down. Done. Martin? Martin, are you there or not? Note it
Yeah,
down or
yes,
not?
sir. Yes, sir.
Note
Yeah,
it down?
yeah,
Okay,
yeah.
great. So please note down. This grouping is based on reaction of bacteria. This grouping is based on reaction of bacteria to grams stain, to grams stain. Full stop. Bacteria are stained first. Bacteria are stained first with weakly alkaline solution, weakly alkaline solution, crystal violet or gentian violet, crystal violet or gentian violet. Full stop. All of them pick up, all of them pick up blue color. Full stop. Then they are treated with 0.5% iodine solution, followed by washing with water and absolute alcohol, water and absolute alcohol or acetone. Full stop. The bacteria which retain blue or purple color, the bacteria which retain blue or purple color called blue or purple color called gram positive bacteria, and those which loses color, those which loses color are called gram negative bacteria. So this was all about the gram staining, and put it as very important question. This is very important for your school exam. Very, very important. Now, this was the story behind the prokaryotic cell. By the way, we have already done prokaryotic cell in Monera part, right? We have discussed in detail about the bacteria. Now we will study about the eukaryotic cell. So our type of the cell as a eukaryotic cell, structure of cell. In eukaryotic cell, we can take the example of any plant cell. Here I have taken example of plant cell. Why I have taken the example of plant cell? Because in animal cells some features are missing, but plant cell have everything, right? We will discuss plant as well as animal cell. We'll discuss about the both of them. Something are missing in plant. So, and you are already aware with this eukaryotic cell structure, but still, look, the in plant cell there will be cell wall outermost covering, which is absent in animal cell. Plasma membrane, the second layer, which will be found in both plant as well animal. Cytoplasm, this is gel-like matrix, ground substance found in both. Central vacuole, this is big vacuole. Try to understand. This is big vacuole found only in plant cell, right? Only in plant cell, not in animal cell. In animal cell, they are very minute and non-functional. In plant cell, the vacuole is the biggest organelle. That's why the nucleus is not found at the center. In plant cell, nucleus are always found at the side, at the corner part or the peripheral part. Why? Because central vacuole occupy a larger size and it pushes this nucleus at aside. Then cytoskeleton, then chloroplast. There are some plastids, peroxisomes. We'll discuss what they are. Mitochondria, it generate energy. Chloroplasts are found only in plant cell, not in animal cell. Plastids in plant cell only, not in animal cell. Mitochondria, Golgi apparatus found in both. Ribosomes found in both. This is the nucleus, second largest organelle, found in all the cells except few exceptions. These structure called endoplasmic reticulum. So we'll discuss about them. And this was all about the eukaryotic cell. So we will discuss one by one what are the structure of eukaryotic. So in your notebook, you have to draw the diagram of eukaryotic cell. And let's start one by one the organelle or part of the eukaryotic cell, right? So you have to put the heading parts of eukaryotic cell. Parts of eukaryotic cell. This is the first part of eukaryotic cell. The first part is that is the cell wall, right? So try to understand. Cell wall is found only in plant cell, not in animals. Look, this is the one cell, this is one cell, and this is the second. Between two cells, so between the two cells, two plant cells, there is a cementing material which work as a fevicol, which keep these cells stuck with each other. It work as a glue, right? This structure is called middle lamella. Try to understand middle lamella. Middle lamella is made up of a material called calcium pactate. So two cells in the plant, they are attached or adhere with each other with the middle lamella. After middle lamella, there will be cell wall. This is primary cell wall. Then after cell wall, there will be plasma membrane. And after plasma membrane, some material get deposited, right? That may be, this is taken like, look, first cell wall, primary cell wall. After primary cell wall, sometime there are secondary cell wall. So secondary cell wall and primary cell wall, it is made up of same composition. Secondary cell wall formation takes place at maturity. Then there is a plasma membrane. After plasma membrane, sometime some material get deposited inside the cells. This is the lignin, suberin, and cutin. When deposition of lignin, suberin, cutin takes place, most of the time cell die, right? So normally lignin, suberin, and cutin do not deposit. Look. So what is the cell wall and how does it? What is the property of cell wall? Cell wall is semi-transparent, right? Its thickness is 0.1 micrometer to 10 micrometer. This is non-living but metabolically active and grow, right? One more important thing that when we talk about the two plant cell, so two plant cell, the protoplasm of two plant cell remains interconnected with each other with the help of some channel. These channels are called plasmodesmata, right, in plant cell, and these channels are called cytoplasmic interconnection or junction in animals, right? That is the difference in both. Both are the same thing. So functionally both are the same, but the name are different, right? Tell me, is it clear or not? Both of you, Martin and Alia. Is that clear? Martin, clear? Okay.
Yes, sir.
So I want you to draw this diagram, just one diagram. Draw this one, this first diagram, this diagram. Make 3 layer: primary cell wall, secondary cell wall, and plasma membrane. And draw these 2 diagrams: plasmodesmata and cytoplasmic interconnection. These 2 diagrams. Quickly. Noted? Okay, fine. So please start noting now.
Yes,
Cell
sir,
wall.
I'm not done.
Okay, Martin. Do it and then let me know.
Done, sir.
Start noting down. Cell was first seen by Robert Hooke in 1665. Cell was first seen by Robert Hooke in 1665. Full stop. It is semi-transparent. Full stop. Its thickness its thickness varies from its thickness varies from 0.1 micrometer to 10 micrometer. Its thickness varies from 0.1 micrometer to 10 micrometer in length. Sorry, thickness 0. Not length, 0.1 micrometer to 10 micrometer. That's it. Next point. Cell wall is non-living extracellular secretion. Cell wall is non-living extracellular secretion or matrix of the cell. Full stop. It is metabolically active and capable of growth. It is metabolically active and capable of growth. Next point. It protects cell. It protects cell against mechanical injury, comma, attack of pathogen, and provide shape to cell That's it. So this was all about the cell wall. One more thing, which is important. The cell wall, the composition, right? The cell wall is composed of matrix. Cell wall is composed of matrix, microfibril, and other ingredient. Full stop. Matrix is made up of water. Matrix is made up of water, hemicellulose, plus pectin plus lipid plus protein, right? So matrix is made up of water, hemicellulose, pectin, lipid, and protein. Microfibrils are made up of cellulose. Is that clear? So this was all about the cell wall. Is that clear? Martin, clear or not?
Yeah, yes, sir.
Great. Now plasma membrane. For plasma membrane, look, give me one second. Let me introduce you. I'm trying to introduce some diagram which you can draw at your school because this is a bit difficult. Not difficult, but maybe difficult for you. Look, this is the diagram of plasma membrane. So basically, a plasma membrane. So second layer is plasma membrane. Every organism have plasma membrane. Either it's plant, fungi, bacteria, or anything, right? It will have plasma membrane. So what is the plasma membrane? Basically, plasma membrane of made up of these two layers. So this is one layer, right? And this is second layer, right? What are these? These are two layer of lipid. Two layer of lipid. So generally we call bi-layer of lipid, right? So this is made up of bi-layer of lipid. And one more important thing, as protein is not for neither found in layer nor found in some structure, it remains scattered. So this is the protein. So protein which are found at the peripheral part means outer regions externally, they are called peripheral protein. And those protein which traverse across the membrane, right, completely from up to down, right, they are called transmembrane or glycoprotein. These transmembrane protein or glycoprotein, they are also called, right, intrinsic protein. What we call them? Intrinsic protein. So intrinsic protein are those proteins that traverse across the what? Membrane, right? Lipid bilayer. Peripheral protein are also called extrinsic protein. Extrinsic protein. This diagram you have to draw if you get. Look, if protein have these sugar carbohydrate chain, we call them glycogen, right? And cholesterol, lipid, these are the ion channels, right? And there are some protein, there are some these kind of structure, they are called ion channels. So they have the core. So this core some ion like sodium, chloride, potassium, they can come inside and they can go outside, right? So they are meant for exchange of material. Is that clear? So this is one more important. This is lipid bilayer, right? So the lipid is this lipid molecule. This diagram you have to make. That's why I'm telling you. This is lipid molecule. Lipid molecule. So this lipid molecule is made up of what? This head and this is. So this head part, this is uh, head, and this is tail. Tail is made up of hydrophilic part, and this tail part is hydrophobic. Hydrophobic. What does it mean by hydrophilic and hydrophobic? This is water loving, right? And this is water repellent. So they are these parts, they are the hydrophilic and hydrophobic. Is that clear, Martin and Alia? First, please draw this diagram quickly. Do not draw this ion channel and all that. At least draw the half of the diagram. Bilayer of lipid, intrinsic and extrinsic protein. That you need to draw. But if you are quick one, you can draw the ion channel as well. Done, Done? Martin? Alia? Hello, Martin?
Yeah, done, sir.
Okay, very good. So, look, one more thing. The term plasma membrane was given by Nageli and Kramer. The plasma membrane is also known as the plasmolemma, plasma membrane, biomembrane, or cell membrane. They all are the same, right? So start writing now. Cell membrane or plasma membrane or plasmolemma or biomembrane are same thing. Are same thing. Full stop. It discovered by it was discovered by Schwann, S-H-W-A-N, Schwann, S-H-A-W-N-N, Schwann. It was discovered by Schwann in 1838. In 1838. Full stop. All membrane are basically all membrane are basically made up of all membrane are basically made up of bilayer of lipid, bilayer of lipid and protein. All membrane are basically made up of bilayer of lipid and protein. Full stop. The average thickness of membrane, the average thickness of membrane varies from 50 to 100 angstrom. Varies from 50 to 100 angstrom. Next point. It is selectively permeable. It is selectively permeable. It is selectively permeable for solute, but semi-permeable, but semi-permeable for water, but semi-permeable for water. If it get injured, if it get injured, is repaired within no time. Means it have a good capability of regeneration, right? The look, one more very important thing. The structure which is visible to you, the structure of cell membrane. This is called fluid mosaic model, right? So this is called the diagram fluid mosaic model. So fluid mosaic model says that lipid is present in the form of fluid and protein swim in this fluid like mosaic, right? Fluid mosaic model. It was given by Singer and Nicholson. It was given by Singer and Nicholson in 1972. Singer and Nicholson in 1972. Right? Lipid molecule have hydrophilic and lipid molecule have hydrophilic and at outer side and hydrophobic tail have hydrophobic tail towards center. Next point. Protein may be peripheral. Next point. Protein may be peripheral or integral. Some protein, next point. Some protein which run throughout lipid bilayer. Some protein which run throughout lipid bilayer are called tunnel protein. Are called tunnel protein or transmembrane protein or transmembrane protein. Tunnel protein or transmembrane protein. These tunnel protein help in exchange of ions. This tunnel protein help in exchange of ion are also called ion channel. Right? Next point. The function of plasma membrane is protection comma, maintain flow of material, and help in cell metabolism, right? So this was all about the plasma membrane or cell membrane, right? Come to the next one. Protoplasm. Uh, just a minute. What is protoplasm? Fine. So try to understand this, Martin. By the way, this chart is for the medical only. So you have to note it down certain things, not all everything. Martin, try to understand what I am explaining. Cell
Yes,
is
sir.
made up of two: cell coat and protoplasm. Cell coat means covering of cell. Now you know that cell coat is made up of, look, the cell coat is made up of two things in cell covering. It may be in bacteria, it may be glycocalyx, right? Bacterial cell and some animal cell. Then cell wall. Cell wall is found in plant, fungi, and bacterial cell, right? Then protoplasm. So everything which is filled inside the cell wall, that is cell membrane, that is a protoplasm, or cell wall, that is protoplasm. This is living. Protoplasm is divided further in three parts: cytoplasm, cell membrane, and nucleus, right? So cytoplasm we will study, cell membrane I have explained, and nucleus I will explain, right? So you have to note down till here. This one: glycocalyx, cell wall, cytoplasm, cell membrane, cytoplasm, cell membrane, and nucleus. Martin, note down, please, quickly, and leave the rest of the part. Note it down, Martin. This upper one.
Which one should I note down? Which one, sir?
This one. Note it down. This one. Glycocalyx, cell wall, cytoplasm, cell membrane, nucleus. Till this point. Note it down.
No, no, I'm doing, sir.
Oh, come on. I'm waiting for you. Look. First, write down cell coat, protoplasm. Then glycocalyx. Leave the rest part. Cell wall, cytoplasm, cell membrane, nucleus. First, please note down this stuff quickly. I thought you must have noted down.
Yeah, done, sir.
Done. Okay. One more thing which you need to note down. Cytoplasm, try to understand. Cytoplasm further divided two parts. Endomembrane system, right? Endomembrane system. Endomembrane system means this is the folding of membrane, right? So cytoplasm, you have to write down that cytoplasm into two parts. Endomembrane system and cytoplasmic structure, right? Endomembrane system means that is the folding of membrane. Golgi body, endoplasmic reticulum, lysosome, and vacuole, right? Then come to the cell organelle. Then further it will be cell organelle and cell inclusion, right? What is the cell organelle? Cell organelle, chloroplast, mitochondria, oh, 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 oh. No, that is wrong. Look, Martin, do like this way. Then protoplasm is made up of two. Endomembrane system. Endomembrane system are made up of folding of membrane. Folding of membrane. The part of endomembrane system are they are first Golgi body, second endoplasmic reticulum, third vacuole, and fourth lysosome. Then there are cell organelle. Cell organelle. So out of this uh, cell organelle, first this will be chloroplast, second mitochondria, then uh, third ribosome, and if this is already there, what else is there? Mitochondria is done, last that is done, ribosome that is done. That's it. So please note on that. Done, Martin?
No, sir.
Martin, done? Or still doing?
Yeah, yes, sir.
Should we go ahead?
Yeah, yes, sir.
Okay, fine. So put the heading endomembrane system. First heading endomembrane system. Put first heading endomembrane system. Endomembrane system is grouping of some membrane organelle, grouping of some membrane organelles, function in close coordination with each other. Close coordination with each other. Full stop. They are following. Put heading A, endoplasmic reticulum. So first, let me tell you what is the endoplasmic reticulum, what is their function. So look, endoplasmic reticulum, it is considered, Martin, this is considered by some of the scientists that endoplasmic reticulum, they are made up of the folding of the nuclear cover or nuclear membrane. So folding of nuclear membrane, it makes the endoplasmic reticulum. Endoplasmic reticulum, if at the surface of endoplasmic reticulum, some ribosome get attached, they are called rough endoplasmic reticulum like this way. This is rough endoplasmic reticulum. If they are just like this way, there is no attachment of any ribosome and all that, they are called smooth endoplasmic reticulum. So rough endoplasmic reticulum and smooth endoplasmic. The smooth endoplasmic reticulum, their function is they produces lipid, steroid, hormone, and synthesize them, right? Rough endoplasmic reticulum, they have ribosome, they synthesize enzyme and protein synthesis, right? They were discovered by Porter and Thompson in 1945. This is the structure of endoplasmic reticulum. This is nucleus. This is nuclear membrane, nuclear envelope, and the folding of this one is called endoplasmic reticulum. So, and this ribosome, uh, this ribosome remains attached at the outer surface, right? And the space inside the endoplasmic reticulum, this is called lumen of endoplasmic reticulum, and the rest of the thing. First, please note down this one: SCR, RR, Porter and Thompson, and this is structure. We have to draw only this structure, only this one. Draw only this structure, only this.
Done, sir. Done.
Done. Okay. So please note down. It was discovered independently. It was discovered not. They have not discovered with each other. It was discovered independently by Porter and Thomson in 1945. Full stop. At places, next point. At places, it is connected with. At places, it is connected with plasma membrane. It is connected with plasma membrane as well as nuclear envelope. Next point. Normally, it forms 30 to 60% of. Normally, it forms 30 to 60% of membrane system of cell. Full stop. They are found quite abundantly. They are found quite abundantly in metabolically active cell. They may be of two types, RER and SER. They may be of two types, RER, is rough endoplasmic reticulum, and SER. Next point. RER bear a fine pore, in the area of fine pore in the area of attachment of ribosome. Full stop. They help in for synthesis of protein, enzyme, and lysosome. Protein, enzyme, and lysosome. Full stop. SER do not have ribosome in their outer surface. They are responsible for formation of lipid, steroid, lipid, steroid, and some hormone. Lipid, steroid, and some hormone. Clear? What is clear?
Yes, sir.
Put the heading Golgi. In next class, I will start from the Golgi body.